Alrighty. Okay, cool. So like I said, this is the last day of brand new content for the year. We will do an activity with this stuff tomorrow, and then we will get situated in the end of the semester. Before I get started, does anybody have any questions about class content yesterday? Anything? All right. So we have talked about how a cell grows, how a cell divides, how a cell knows when to grow and divide, and how a cell, or what happens to a cell when they don't listen to those signals. But what we haven't touched on is how a cell knows what type of cell to be. So we have lots of different types of cells in our bodies. We have skin cells, we have heart cells, we have liver cells, we have blood cells, all these different types of cells. We only have one set of DNA, right? Like the DNA in our brain cells is the same DNA in our skin cells. But how do we know what type of cell that a cell will become? And we'll talk a little bit more about gene expression when we get into genetics next semester. But today, we are going to talk about cell differentiation. This could also be called cell specialization. The main big overarching question you should be able to answer by the end of class is how cells become specialized for different functions. How do cells become specialized for different functions? Our liver cells have to be really good at detoxification. That's what our liver does. But our skin cells have to be really, really good at protection. So it all starts with development. So at the very beginning, right, sperm meets egg comes together, that becomes a zygote. And as that zygote develops into a couple more cells, it gets to be a clump of cells, we call that an embryo. So as we develop, we go from one cell to an embryo. An embryo is the developmental stage where cells start to become differentiated. An embryo is the developmental stage where cells. What's wrong with my handwriting today? So way back when we were but clumps of cells, we started to become specialized. So our body kind of had a plan of what cells were to become bone cells, what cells were to become brain cells, just a general guideline as to where certain cells need to be in order to form a fully functioning human. I've used the word differentiation a lot. So let's put a definition to that term. Differentiation is the process by which cells become specialized for life's different processes. Now, I feel like I've used the terms specialized and differentiation to kind of describe one another. Is everybody doing okay with where we are so far? I get a thumbs up, thumbs down, thumbs in between. Okay. So, differentiation is the process by which cells become specialized for life's different processes. The process by which cells become for life's different functions. This is a really, really, really stinking cool process. I spent half a year in college just studying this stuff. And it was an absolute blast. One of my favorite classes I've ever taken. And what's really cool about it is even though all living things start as one cell, right? In some organisms, differentiation looks different than in others. So, in some organisms, like this, there's a worm that's pretty common in like the field of biological study. So they're like we used to use lab rats a lot. Now we use these worms. It's called C. elegans. So in some organisms, like those little worms, cells' roles are strictly determined by a specific point. So we get to a specific point in development. I think it's like a day or two in. And they're like, okay, our cells know exactly what they're going to be and where they're going to be. So cells on this layer of this ball of cells are going to be the nervous system. Cells in this layer are going to be the digestive system, and so on and so forth. So in some organisms, like our worm friends, cells' roles are strictly determined at a certain point in development. So, C. elegans is pretty common. We know a lot about how this worm goes from a single fertilized egg to a big old worm. I guess it's not big. But other organisms, like you and me, like mammals, go through a more flexible process where there's a bunch of different factors that come together to control the process of differentiation. Um, so it's all about what signals are produced and when those signals are produced. So in other organisms, it's a lot more flexible. Other organisms, like humans, this process is more flexible. Which is where we're going to spend the rest of this kind of lecture. Because while C. elegans are super cool, we care a little bit more about how humans develop than how these worms develop. So we're going to talk a lot about human development. How we get from a fertilized egg to an embryo to eventually, possibly, a human. And how those cells come together to make different types of cells. How we get from one fertilized egg to skin cells and eyeball cells and lung cells and the like. So, In humans, the very, very, very first cell is called a zygote. Zygote. A zygote. That is the first cell of a human. If a sperm meets an egg, they combine. That one cell is called a zygote. That cell has the potential to become a human. Um, or I guess that's how most mammalian development works, but we're just talking about humans. So that first cell is called a zygote. Zygotes are what we call totipotent. Zygotes cells are totipotent. So a totipotent cell is a cell that has the potential to become anything. So when a, a fetus is growing, is developing, from this one cell, we are going to get all of the cells that make that embryo, and we are going to get all of the cells that make the placenta that wraps around and protects that embryo. So, totipotent cells mean that they can become anything. That is body cells or placenta cells. So that one cell is a zygote. It is considered to be a totipotent cell. Totipotent cells can become anything. They are, if that zygote is to continue to grow, will become a fetus and the placenta cells. But after about 4 days, that zygote has grown into something called a blastocyst. A blastocyst is a hollow ball of cells with a cluster of cells inside. So the outside of that ball of cells will become the placenta, and the inside of that ball of the cells has a potential to become a body. So after about 4 days, the zygote becomes a blastocyst. A blastocyst is like I said, a hollow ball of cells with a cluster of cells inside. So our zygotes are totipotent. They're going to become the outside of this hollow ball and the inside of this hollow ball. The outside of that hollow ball will grow to become a placenta that will nourish whatever fetus grows out of the cells that are in the inside of that ball. Cells in this cluster are considered pluripotent. So the cells on the inside of that hollow ball are pluripotent. That means they are able to become any body cell. They have lost the ability to become placenta, but they can still become any body cell. So any cells in this inner cluster, if exposed to the right chemical signals, can become anything from a heart cell to the cells in your big toe. Doing okay so far? Okay. So these cells in this cluster, these pluripotent cells, These are unspecialized cells from which differentiated cells develop. They are called stem cells. Show of hands, how many of you have heard or interacted with the word stem cells before? Okay, yes. So stem cells are unspecialized cells from which differentiated cells develop. It's a little wonky. So we have become a blastocyst. That blastocyst is the outer cells and inside of this and the cluster inside. The cluster inside are pluripotent stem cells, which means they can become anything, any type of cell in our body. So there are two main types of stem cells. So, two main types of stem cells. There are adult stem cells and embryonic stem cells. Embryonic. Yeah. So, embryonic stem cells are the stem cells that we talked about inside of that blastocyst. Adult stem cells are the stem cells that still exist in our bodies. We have stem cells in our bodies right now. I'm going to talk about the difference. So, adult stem cells are a little bit more restricted. So adult stem cells are found in little clusters around the body. Some examples are, let's see, bone marrow, hair follicles. There are some in the heart and some in the brain. These adult stem cells don't necessarily have the same potential that embryonic stem cells have, so they are considered multipotent. Multipotent stem cells are limited to replacing cells in the tissues where they are found. Limited to replacing cells in tissues where they are found. Blood cells are a really good example of this, right? So we have lots of different types of blood cells. We have red blood cells, we have white blood cells, we have platelets, all that fun stuff, right? So the stem cells found in our bone marrow are in charge of making both bone cells and blood cells. All of our blood cells are made from these multipotent adult stem cells that are found in our bone marrow. So our blood cells don't really replicate on their own very often. Instead, they come from these stem cells that are in our bone marrow. Yes. I guess. So, those are adult stem cells. We also have embryonic stem cells. Embryonic stem cells. And like I said, they are pluripotent. So we have that first zygote cell that was totipotent, meaning it could be anything from the cells in the embryo to the cells in the placenta. We have embryonic stem cells in the middle of that blastocyst, which are pluripotent, which means they can form any of our body cells. And we have adult stem cells in places like our bone marrow and our hair follicles that are multipotent, which means they are limited to replacing cells in tissues where they are found, but they can become a bunch of different types of cells within that tissue. Doing okay? Okay. Now we get to talk about some real life applications of this stuff with stem cell research. We're not going to get too deep into the ethics of it all. Just kind of laying about. So, stem cells are really, really, really intriguing to scientists because if they have the potential to become any type of cell, there's a lot of ways for us to study development and how those cells get to be from one type to another. And it can help us develop treatments for diseases where our cells aren't growing as fast as they should be. So, the potential benefits for stem cell research. Potential benefits for stem cell research. So, it makes it possible to develop new fields of regenerative medicine. Develop new fields of regenerative medicine, where we can use undifferentiated cells to replace damaged cells. So there are degenerative disorders where people's nerve cells break down over time, where people's heart cells break down over time, where people's muscle cells break down over time. And scientists believe that they can use stem cells, they can use these cells that can become anything to help regrow or at least help regain some abilities where people had lost them because of these degenerative diseases. But with this comes some ethical issues. Less so with adult stem cells because they can be donated. We can get permission to collect adult stem cells. And this happens when people do bone marrow transplants. So, one of the biggest treatments for leukemia or cancer in the blood cells, right, is to get a bone marrow transplant. By taking the bone marrow out of one person and putting it into the other, we are essentially transferring adult stem cells from one person to another. And those adult stem cells in that bone marrow will help create blood cells for a person who has lost a lot or has a lot of damaged blood cells. Embryonic stem cells get a little bit more dicey because we've got arguments for and against it. And when people argue for and against it, it'll come down to a argument of life and death. Meaning, if we are harvesting embryonic stem cells, we are most of the time destroying embryos, and that kind of brings in a lot of ethical issues. So, embryonic stem cell harvest involves destruction of embryos, I forgot how to spell embryos, which involve ethical issues of life and death. So, embryonic stem cell research doesn't happen very often because when it does, it is often a very big controversy. So, without being able to use embryonic stem cells in these regenerative fields, scientists think that they can't get as far as they need as fast as they need. But science finds a way, and there is now a way around this idea of controversy. I'll get that down. I realize I wrote a lot. So there's a way that scientists have found to do research with pluripotent stem cells without the destruction of embryos, without the creation, without the use of embryos. And that has to do with cellular reprogramming, which is super stinking cool. Cellular reprogramming. So, by studying genetics, by studying development, scientists have found a way to introduce factors to cells that gets them to revert back and become able of reproducing other types of cells. They were able to take, I think it was fibroblasts, so like a type of skin cell, introduce it to a bunch of factors and get things like heart cells and liver cells out of this reprogrammed cell that all they did was expose it to certain factors. So, by studying genetics and cell signaling, we've talked about cell signaling already. We're going to talk about genetics next semester. By studying genetics and cell signaling, scientists can introduce factors to differentiated cells and cause them to reproduce other types of cells. This was a really big breakthrough. This is a really, really big deal. By being able to do this, we have opened up so many doors in regenerative medicine. If we are able to take something as simple as a skin cell, introduce it to a bunch of factors, a bunch of cell signals, and get heart cells out of it. That's insane. That's so stinking cool. So we call these cells, or these cells that have been converted to cells that closely resemble embryonic stem cells, induced pluripotent cells. These converted cells are called induced pluripotent. Because they are pluripotent, we can get specific types, or we can get all of our body cells out of these cells with the right signaling factors. But they didn't start out that way. So we call them induced pluripotent cells. These induced pluripotent cells allows research to take place to create life-saving treatments without taking stem cells from embryos. Allows life-saving research to continue without the destruction of embryos. So now scientists don't even have to think about the ethical issues or the ethical backlash from their research, because they can just make their own induced pluripotent cells without having to harvest them from a human embryo. Anybody have any questions? This stuff is complicated, but it's super, super cool, and I love talking about it. All right. That concludes the end of new content for the semester. So everything we do from now on is going to be a review. Tomorrow we'll do an activity with cell differentiation, and then we will kind of glide into the end of the year. There is an exit ticket on Canvas for you to fill out, please, and thank you. It is your last exit ticket of the semester, which is super great. Y'all are killing it. Let's see, where are we at time wise? Perfect. You have about 10 minutes to get that done, knock that out. If you finish it, why don't you go back through your Canvas grades, make sure that you have answered all of the other exit tickets for the year, just because those are for the 9 weeks. Because those might only be 5 points today, but those add up over time. So, and if you have any questions about this, if you have any questions about your project, if you have any questions about other missing assignments, please let me know. As we are approaching the end of the year, you gotta advocate for yourself as opposed to me hunting you down all of the time.